Yeah, we are very happy to host Dr. George Constable. He is an assistant professor in the Department of Mathematics at the University of York. Before in York, he did a postdoc in Princeton and then in Zurich, and also spent some time in the University of Bath, I think, right? Yeah. His background is mathematics and physics, right? And he's working on evolution and ecology. So it would be lovely to hear from him. The stage is all yours.
Lovely. Thank you. And thank you very much for inviting me. And I have the impression from talking to people that this audience is unusual, so I hope I pitched this correctly. So who is a mathematician or a physicist? And who is a biologist? And who is something else? What is something else?
Psychologist.
Psychologist. Okay, cool. So I'm not sure I'm prepared for psychology. So, but feel free to interrupt as I go along. I've tried to kind of pitch this, from alternative directions. So, but please, interrupt whenever you have a question. Okay, so just to kind of ground us, I'm interested in sort of stochastic eco-evolutionary modeling. And so kind of, I might think about modeling something. I might sort of initially think I want to potentially model this with an ODE. So I could start with something like logistic growth here, where I have dx by dt is Rx minus Cx squared, where C is some competition rate, and I get my nice sort of simple curve. And obviously for more complicated models, I can just generalize this to some vector x and see how that changes in time, dependent on this vector valued function a of x, okay? But I might also want to model that sort of stochastically. And if I wanted to do that, rather than thinking about an ODE, I might start thinking about the probability per unit time of certain events happening. So for instance, by the way, if anyone can't see anything, I've realized some of these things might be a bit small. But yes, so I might have, instead of considering this kind of continuous quantity x, which tells me something about the abundance of individuals in the population, I might consider a discrete thing, which is this small n, and I might say, what's the probability per unit time that this discrete number of individuals is going to go up? And that's t of n plus 1 given n, and that would happen at a rate r times small n over big N, where big N here is measuring something about the environment's, the volume of the population, or the environment's ability to contain a certain number of discrete individuals. Okay, and similarly, I could say, what's the probability per unit time that I lose an individual stochastically? And that would be given by C n squared over n. And importantly, I can map, as I take n to infinity, right? I can map from these probabilities to this ODE. And again, for kind of a multivariate system or a more complicated model, I can also approximate these types of probabilistic models by an SDE rather than an ODE. So I have dx by dt equals a of x just like before. So I have some deterministic component, but now I'm adding some extra kind of noisy bits on the end. So some random kind of kicks essentially. And the strength of these kicks might depend on my position in state space, which is this g of x here. So I can simulate all of this kind of straightforwardly. So here I've got gray lines representing sort of my stochastic trajectory. And if I add noise, I just get some wiggles around my lines. I can reduce my population size. And if I reduce my population size, then I get more noise, okay? Because I have a fewer number of individuals, so there's more uncertainty in the ordering of these birth-death events. I end up with more noise, okay? Okay, so smaller population sizes lead to more noise. And that's kind of obvious from this SDE, okay? So there's this noise term here, and I find this term at the front, which is 1 over square root n. So as I reduce my population size, the effect of this noise gets amped up, and I end up with these kind of more stochastic looking trajectories.
Sorry, the x1 is the total, the average population. What is it?
Pardon?
What is this x variable meaning?
Oh, x. So x1 here is, yeah, so x1 here is just x. So this is just logistic growth. I've got some variable x that grows in time. And the black line is saying, I can look at an ODE for that. And the gray line is saying, okay, what if I simulate this kind of equivalent stochastic model? and do some scaling. And if I simulate the stochastic model and I have smaller population sizes, then if there's fewer individuals in the population, basically there's more uncertainty because I'm kind of averaging over a smaller number of individuals. And so I get these kind of wider fluctuations away from this deterministic prediction. Is that okay?
Yeah.
Okay, cool. And but of course, if I average over all the stochastic trajectories, I get this red dashed line. And so that's kind of a bit boring because that's just basically I've gone to all this extra effort of adding all this noise in and I just get this sort of smear and who cares? So more interesting is when I have a very noisy system, so say a very small population size, because then I start to get deviations from potentially of my average of my stochastic trajectories from my deterministic trajectory, okay? And this is kind of a trivial example because all that's going on here is that I have extinctions that are possible in my stochastic system but aren't possible in my deterministic system, and they're basically pulling down this average. And it's trivial, but it's basically just to kind of sort of emphasize the point that if in large noise systems, I might start seeing departures between the mean and the deterministic behavior. Okay.
Here's the statistic predictory. Do you mean the discrete one or the SDE one?
Both.
But for the SDE one, whether or not you see extinction really depends on the structure of your G. But
yes, so I'm thinking about this SDE. I'll get to that. So I go through this not a bit more formally, but a bit more carefully in a minute. I'm thinking about SDEs that are derived from the individual-based model, which is formulated in terms of these discrete transition rates. But I'll get to that in a. But yes. So yes, this G of X, the form of this is particularly important.
So can I ask you?
Yeah.
When we have very small populations, we get this, yeah.
Yeah.
How small are we talking about? Because a rule of thumb is something like if you have more than 30 or 100 individuals or something like that in that order, then you are basically you can do a deterministic approximation.
Not always.
Fine.
Yeah, not always.
Not always.
Well, it depends what. Well.
Yeah. So do you have any idea about like what numbers are we talking about when you say very small populations?
No, because it depends.
Okay.
It depends on all the other. So it depends small relative to what, because it depends on all the other parameters basically. Okay. Because I'm going to look at some purposefully extreme edge cases where the answer is these noisy effects dominate for whatever population size you look at, but that's only true because I've looked at an edge case potentially. Okay. So in principle, whatever size I want, but I'm kind of, yeah. Well, we'll get. Is that everyone okay with that? Okay. So in terms of my research interests, I'm not really interested. So I'm interested in when does stochasticity lead to qualitatively different behavior, because the picture that I showed earlier where we just end up with some wobbles around the mean is less interesting. Why go to the extra effort of looking at stochasticity if it's not doing anything fun? And kind of related to that, I'm also interested in when does stochasticity, or adding this genetic drift essentially into these models, help provide a parsimonious explanation for empirical data. So essentially, when can I take a theory that involves a large number of variables with potentially complicated functions f1 to fn here, and kind of replace it with a lower dimensional model where hopefully these functions G1 and G2 are a bit simpler, sort of embed fewer modeling assumptions, but by just adding some kind of realistic noise on top of it, I somehow recapitulate the same observed patterns, okay? And so neutral theory would be kind of that type of thing, right? So if I think about Hubble's neutral theory, you take really complicated model, ecological model, and you replace it with something that's basically just noise, and you say, okay, that gives us some observed distribution. And that doesn't mean that it's the right answer, but it does tell you something about the quantity that you're looking at. In that case, species abundance distributions. I'm also kind of interested in this question of when can we actually leverage this noise to tell us more about a system? So if we propose a model, and that model then comes along with some predictions about what the form of this noise should look like, can we use that noise with real sort of biological noisy data to tell us something about the system and what's going on? And completely separately, I've gotten interested in facultative sexual reproduction. So that's most microorganisms reproduce asexually or clonally most of the time and only engage in sexual reproduction in stressful conditions. I got into that kind of problem because it's a very noisy system and I'm interested in noise. But now separately, I've just got interested in the biology. But I don't have time to talk about that today, so I'm just going to focus on kind of these two. But if anyone's interested in the weird sex lives of microbes and fungi, I'd happily talk to you about that later.
Just one question. So are you going to control the noise or are you going to derive the noise from some micro model?
You derive the noise from micro model, yeah. And that, yeah. Okay, so then today's talk is going to be quasi-neutral Lotka-Volterra models because everyone knows a Lotka-Volterra model and they're kind of straightforward, but they kind of give you some intuition as to what's going on. Then a brief foray into sex chromosome evolution to show that this isn't all just kind of too artificial, and then some models of cellular growth, coalescence, and fragmentation. Okay, so what I'm going to do is I'm going to start off with a master equation. So this is just what is the probability of being in some discrete state n at time t. So n here is a vector. So it tells me how many discrete numbers of individuals I have in each class. And the master equation or the master equation is basically telling me what's the probability, how this probability of being in state n changes in time. And it's kind of intuitive sort of to describe the probability that I'm in, the probability of being in state n at time t changes with the probability that I was in some other state n prime, some other state n prime, and that I transition from that state n prime into state n. So this is the probability, this t n given n prime is the probability per unit time of transitioning from state n prime into state n. And the probability of being in state n at time t simultaneously decreases with the probability that I was already in some state n and I transitioned out of that state n and into another state n prime. And I just sum that over all the possible transitions, and that tells me how my probability distribution evolves. Okay. Any questions about any of that? All good? Yeah, okay. So then, obviously, to encode the model, I need to specify what these transition rates are. So if I'm looking at sorry, this is all a bit small. If I'm looking at it, okay, it's a bit small, so any questions, I can't read anything, do shout at me. So the probability of some species xi increasing in time, so a birth event is given by this parameter bi. The probability per unit time of a death event, so this xi going to nothing is di, and the probability that a type I and a type j interact leading to the death of a type I is Cij. Okay, and the master equation, straightforward enough to explain what it is, but nightmare to deal with mathematically in this situation. So I'm going to use a Cramer's Moyalic. So if you're not interested in the maths, this is just how I get from now to an SDE. If you are, I'm just explaining how doing all this. Use Cramer's Moyalic expansion, so just a nonlinear version of a Van Kampen system size expansion. Basically approximate these discrete number of individuals, ni, by a continuous variable, where ni is where xi is now kind of measuring the density in some sense of these individuals in some volume v. And that allows me to get to a drift diffusion equation, the Fokker-Planck equation, which is basically what I've done between this top step and this bottom step, is gone from looking at a discrete probability distribution to a continuous probability distribution that's kind of easier to deal with. And conceptually, why this is useful is that this is not equivalent, but in some sense, this Fokker-Planck equation is equivalent to this Ito stochastic differential equation. Okay. Right. Any questions about that? Everyone kind of happy? Yes. Okay. Cool. Basically,
Do you know what the form of Bij is?
yes. Yeah. So you can calculate all this is kind of like what you call it, Turing machine kind of stuff. So for any model basically for any model, if you just write down a bunch of transitions within, you can stick it into a computer even to do this because the packages for it, but it's quite straightforward to by hand just work out what Bij and AI are.
But what is it for the local hotel?
It's a diagonal matrix. I can. Is there a pen?
Yes.
It's so for this. In fact, I'll get down if I go to the next page. So this is the SDE you end up with for the stochastic block of Volterra. These are you've got something dxi by dt is ai of x plus this noise term. The deterministic bits are just what you kind of imagine from the block of Volterra, and the noise terms are going to be it's a diagonal matrix.
Is it just Gaussian delta?
Yes, yeah, but no. So you've got are you asking for the structure of Bij as function of x?
Yeah, that's what I was. Yeah, I know it's the matrix, yeah, but
No, yeah. So Bii is equal to xi times Bi, and then there's a plus di. So basically, it's exactly like this, but with all the minuses turned to pluses. So you get Bi plus di, and then plus. Sorry, this isn't
I cannot see everything.
I can see it up close. Is that better?
Yeah, no, yeah, it was just the color.
Okay, sorry. So that's xi, and then it's Vi plus di plus x cii xi squared plus cij xi xj. So it's basically exactly like that, but with all pluses instead of minuses. And if you want an interpretation of that, it's that either a birth or a death event adds the uncertainty in adds the strength of the noise.
Sorry, you don't have special dynamics.
No, nothing
Yeah,
special.
sorry. All of a sudden, no, but yeah, you get Gaussian noise. It's just that you don't have spatial dynamics.
Yes, yeah.
No,
Okay, sorry.
Isn't there a square root of the whole thing, or is it
No, there's no square root because it's so it'd be a square root. It's because of how I've defined B here as the correlation.
the square of the
Yes.
square root? Yeah.
Yes.
Okay. Yeah.
Okay. Any other questions?
Can you say again, so these x1, x2, they represent densities?
Yes. So x1 equals n1, so the number of n1 individuals over some parameter v, and x2 is n2 over some parameter v. And basically, this parameter v, all it's doing is it's controlling the strength of the noise. So large V basically means that for a given x value, my number of individuals is larger. If I mapped from these x's to my discrete number of individuals, and a larger number of individuals means less noise. So it's basically just a way of trying to. This Kramers-Moyal expansion that I mentioned is basically just a way of writing out an expansion in these kind of uncertainties that come from demographic fluctuations. And so essentially what this is saying is if you have an infinitely large population, you're averaging over all of the interactions in an infinitely large population, there's absolutely no uncertainty, everything kind of behaves like it should on average. But if I take a small finite population, then there's some sampling error but a sampling uncertainty in what's going to happen. And that's what V is kind of capturing. That makes sense?
So that means that these xi's must remain non-negative.
Yeah.
Yes, yeah, sure.
And is that guaranteed by this?
Yeah.
Because I can't see this.
Yeah, I'll show you some phase portraits basically. Okay. Oh, and yeah, also the noise, the noise disappears at the. So because all these noises are defined from the individual base model, a natural property of that is at the boundaries, the noises all switch off.
Oh, can I have a question? What is the structure of the matrix A? Is it antisymmetric or is just asymmetric matrix?
So which matrix?
The matrix A's.
A?
Like, sorry, not A, but like C.
C. Oh, C doesn't matter. So I've just kept it general for now.
Antisymmetric or asymmetric in general?
It can be. We'll see a case where it's asymmetric in a second, because I could have one species suffers more from competition. Species one can suffer more from competition with species two than species two suffers from competition with species one.
But then if the Cij is asymmetric, then if I look at the Bij here, right, it's it's also not symmetric matrix Bij.
No, so this is BII.
Bij, so Bij is zero.
Bij is 0.
Okay,
Yeah.
thank you.
In your previous slide, you defined R as T over V.
Did I? Where did I define that?
Yeah. Moyle expansion R is T, tau is T over.
Tau, yeah.
Oh, sorry.
Sorry.
Tau.
I thought
Yeah,
this would be a bit bigger in my head. I was also closer to my screen.
but then you go back to T again, so what's?
Oh yeah. So that's just so I'm
Okay.
being sloppy. I'm
Okay.
being very sorry.
That's fine.
I've just formally rescale time
Yeah,
in this
you rescale
expansion,
time.
but I get bored of writing tag too quickly. Sorry, that's just me being sloppy. So deterministically, you just get your standard things that you can get in a Lotka-Volterra equation. So I can have open circles or unstable fixed points, solid disks or stable fixed points. So I can get selection here for species. If I look at my phase portrait, selection for species X1, depending on my choice of parameters B1, D1, Cij.
So now
Yeah.
I guess you don't want to, you were avoiding simulating the microscopic model, I guess.
Yeah. I'm just saying, where does our intuition? Yeah,
I just wonder what you want to get from this. You just want to get now you can compute 2-point correlation functions with the x's.
No. I've got my SE, and now I'm just trying to build some intuition. And I'm going slowly because I didn't know who was going to be here and how, what level to aim at. So I'm just basically kind of doing a quick recap of what's going on, basically. But I can hurry up over these bits.
No.
Just because it's that thing of not knowing exactly where everyone is in terms of their interests and things like that. But basically, you can get your standard things that you get from a deterministically anyway. You get your standard things that you can get from a Lotka-Volterra model, so you can get bistability or you can get coexistence. But obviously, when we add noise onto this, we've got uncertainty. So this is kind of a histogram of the different sort of probabilistic trajectories that I can go down. And here you can, in the deterministic system, I would have a stable fixed point here, and now I've got this smudge of kind of uncertainty arising from the stochastic system. Okay, and so importantly, even though I've got the stable fixed point here in the deterministic analog, I can see that there's leakage of probability through this extinction boundary.
So the intensity of the color quantifies how many trajectories stay there?
It quantifies how many times a simulation ran over that point.
Okay.
Basically, or visited that point. Okay. So basically, I've got some distribution kind of hovering around. So a little bit like you saw earlier. So I could probably plot something approximately Gaussian hovering around that fixed point. But part of the tail of that Gaussian is sticking out into this boundary. And so I've just got probability leaking away all the time, and so this distribution is collapsing slowly as it leaks away.
But is this the simulation of that equation or of the other model?
This is a Gillespie simulation of the original individual base model. Yeah. I'm just using the SE because the SE is easier to deal with and kind of more intuitive because you can do this.
You're not using it, you're showing it.
I'm showing it
because
it's a nicer way to think about. If you're trying to think about where does the stochastic system depart from the deterministic system, the SE is the nice way to view that because you're like, okay, well that's what the determinist. I basically ignore the last bit. What does that do? Stick it back in. And when I say stick it back in, what I really mean is simulate Gillespie, or do some maths that involves.
And so, and the right-hand case, it's the coexistence case.
So that's a coexistence case in the deterministic model, but basically what this is a coexistence case in the deterministic model, but in the coexistence
Yeah,
model.
yeah, so I understand, it just corresponds to the third.
Yes,
But all I meant to say before is that now you can use this equation to compute the variances.
yes, but I can also, but
Analytically.
in particular, I'm kind of interested now in some, instead of thinking about stable and unstable fixed points, fixation probabilities basically, and fixation quantities is what I'm interested in. So what's the probability that given some initial fraction of type 1, that I end up at this absorbing state down here? That's kind of what I'm interested in. Okay. So for instance, this red dashed line would correspond to something like this deterministic phase portrait. So what this is saying is that if I start off with a large fraction of type 1s, they're selected for, and there's a large number of them, or I start off with a high proportion of type 1s, I'm just going to get sucked here with a high probability, and that's why that red dashed line is near 1. But obviously, if I start down here with a small number of individuals, even though type 1 is selected for in that scenario, it's obviously not going to fixate with certainty. Okay.
It will depend on your integration time. I mean, if you send t to infinity, you will always end up at the absorbing boundary, or
not if I simulate this using a Gillespie simulation. What do you mean? Sorry, yes, I always end up at a absorbing boundary, but this is this is the fixation probability of type 1. So this fixation probability is saying what is the probability that I bump into this boundary where x2 has gone extinct before I bump into this boundary where x1 has gone extinct. There. That's kind of what I'm interested in.
So the 4 lines in that upper panel, I mean, you're now talking only about the one that is hardly visible.
Sorry, this is I thought.
So yeah.
So what's the question?
The question is, like, you're talking about fixation probability, but that's for then corresponding to the lower, and you point out to the lower left panel.
So for instance, so these are just this is trying to this is trying to illustrate a bunch of different possibilities for the types of sort of functions that we might see for the fixation probability. So for instance, this red dash line says I've got a high probability of fixation. It's basically above the diagonal, so I have positive selection for that type. So that corresponds to that case. And just pointing out, obviously, the fixation probability when this initial type 1 is rare, even if it's selected for, is going to be low, because that's how it kind of works, right? And then these are just some other shapes that I can get from this type of system. So for instance, this thing here, this curvy one, corresponds to this fixed point. This is, I have an enhanced probability of fixation. If I start below the coexistence fixed point, I have a suppressed probability of type 1 fixating if I start above this fixed point, because everything's being dragged in. But this is just kind of trying to orient people. And also there's a chance for me to work out which bits people are happy with and which bits are people are.
Sorry, can you just tell me how you compute that probability?
From the backward Fokker-Planck equation. So you take the Fokker-Planck equation, and you basically start manipulating that, and you get take the Fokker-Planck equation, write down the backwards Fokker-Planck equation, manipulate that, and eventually you can solve for fixation probabilities and fixation times from that. But I could also have done other things apart from that approach, but that's one of them, and it works. Any others? I can also calculate fixation times, but I'm not actually going to worry about that, but just thinking about average time to fixation. Okay. Right. Now I want to look particularly at a kind of limit, a weird limit of this. It's because so you're asking how low does V have to be? So let's look at a weird example, which is neutral conditions. So this is, I'm going to set all the B's equal, all the D's equal, and all the C's equal. So basically all of these two types are basically identical, just labeled differently. And then my phase portrait, instead of having these stable fixed points and unstable fixed points, I've now just got a line of fixed points, this center manifold. Okay. So in the deterministic system, the population just grows, hits some point on this line and stays there indefinitely. In the stochastic system, I approach this line. Once I get to the line, so I basically feel a kind of deterministic push to hit this line. along this red stochastic trajectory here. But once I get there, there's no dynamics along the line. And so I basically bounce just entirely driven by noise until I hit one of these absorbing boundaries. Okay. Here, the fixation probability, I can go away and calculate this. And the fixation probability is Q of F1 equals F1, where F1 is the initial fraction of type 1. Okay, so this is just like a Moran model or a Wright-Fisher model from population genetics. This is just my initial frequency, my fixation probability of type 1 is just my initial frequency of type 1, and that's that green line, this green straight line here. Okay. Any questions about that? Okay, right. Okay, so now, that's what I would say. Okay, so that's nice and straightforward.
Okay, yeah, can you explain to me why you say like, okay, my fixation probability is Q of F1 equals F1?
Yeah.
Where do you get that from?
You end up with, I can conduct a change of variables, okay, and I can write this as, I'll actually get to this a bit later, but I can write this as an equation. Instead of looking at x1 and x2, I can write an equation for f1, the frequency about f1, and a separate equation for n, the total population size. If I write it in terms of that, and I make a transformation of variables, what I get is an equation for f1, df1 by dt equals 0, and just some noise, which is kind of what we see, and an equation for the total number of individuals, dx1 plus x2 by dt, and that just collapses to this carrying capacity. You can use fast variable elimination to say, okay, that collapse in to carrying capacity happens very quickly. I'm just going to look at the dynamics of the frequency, which then is decoupled. And these decoupled dynamics for the frequency, I solve that and I just get that. Solving backward focal Planck.
But what you do see is that actually you have variability around the line.
Yeah, doesn't matter though, and I'll get to why in a minute. So it basically everything cancels. Yeah, but that you're right. I'm glad you said that because it says this isn't some of this isn't necessarily trivially obvious. But basically, yeah, this actually just this collapses to the Moran model, but with a population size given by whatever your population size is here. And it's identical to the Moran model after you do that approximation of the. Okay, what if I look at quasi-neutral conditions? So now what I'm going to say is I'm not going to set all my B's and D's equal, but I'm going to set the difference in B's and D's to be equal. So B1 minus D1 is going to be equal to B2 minus D2 is going to be equal to R. And I'm going to set all of my competition rates just the same as before. But now I've got some difference in my birth rates. Okay? So that changes these equations down here. But essentially, well, I've just changed B minus D to R. It hasn't actually changed anything in a substantive way. What?
Well, you've changed.
Yes, but I have changed the noise.
Yeah,
exactly. So deterministically, this phase portrait is exactly the same. Nothing has changed in terms of my deterministic description, but my noisy description has changed. So what happens to type? So if I now say, I've got, sorry, this is also small. I've got a rate R is equal to 1. Okay, so B1 minus D1 is equal to 1. And I've set here B1 is equal to 2. So for my species 1, I've got a birth rate of 2 and a death rate of 1. Okay. For species 2, I can now change B2 and D2. So I could have species 2 with a birth rate of 10 and a death rate of 9, and they still have the same reproductive output, but they have different values of B and D. So if B2 is equal to 10 and D2 is equal to 9, does anyone have any feeling about what might happen? Is the fixation probability unchanged? It's changed, but in which direction?
Lower
For who?
for 1.
So species 1 will have a lower fixation probability. Okay, it actually has a higher fixation probability, which is good because it makes the point of doing some maths, because it's not entirely intuitive. But basically, as I increase, so this is B2 is 100, which is kind of ridiculous, but species 2 has a birth rate of 100 and a death rate of 99. and you see this kind of noise-induced quasi kind of selective behavior emerging. Okay, so the fixation probability, if B1 is less than B2, then it has a increased fixation probability, and this is kind of a drift, genetic drift, or noise-induced selection for this kind of slower lived species. Okay. Any questions about that? No? Okay. So, what's going on? So if you're a biologist, you might think about Gillespie's criterion. So essentially, there's selection for decreased variance in offspring number. If I'm giving birth at a rate 100 and my offspring are dying at a rate 99, there's a huge variance in my reproductive potential. Whereas if I'm giving birth at rate 2 and my offspring are dying at rate 1, then that variability is decreased. Okay, so this is one way of doing it, is viewing it, is the selection for decreased variance in offspring number. So that's 1977, Gillespie wrote that. Okay, but this is just exactly the same thing he described, but with a slightly different mathematical framework. Mathematically, if you're a mathematician, you might think of this in terms of Ito calculus. So this is an Ito SDE, so the normal rules of calculus don't apply. So basically what I find is that if I evaluate this term, I get 0. So deterministically, there's no change in the frequency between these two types. But because I've got this nonlinearity in my frequency dynamics compared to my ecological dynamics, and I have this noise term, and there's this difference in the types in my noise, I get this extra term, and this creates this noise-induced selection. And another way to view it is kind of geometrically. I've now got these asymmetric noise terms for these two species. And essentially what's happening is one of the species, species 2, is kind of experiencing more noise. And you can kind of imagine that I've got fluctuations that are taking me off the center manifold. And then once I'm off the center manifold, I'm being caught up in these deterministic dynamics again and kind of forced back to the line. And if I kind of draw that out, what I see is that there's a kind of bias in the direction of type 1 fixating. Okay, so if I jump up, I get projected back down there. But if I jump down, I get projected over here. And there's a bigger distance here than here. There's a bigger distance there than there. So I have this kind of asymmetry, which is knocking me that way. Okay, any questions about that? No? Okay, right. So,
You can minimize the.
how do I do that?
With your finger?
Yeah.
Oh, okay, sorry. That, like that?
Yeah, and move it around as well if you like. You've got more pictures coming in.
Okay. Thank you. Okay, so now let's look at something that just has weak selection. So I've seen there's this noise-induced selection for the type that kind of reproduces or that has slower life sort of history characteristics, which gives them lower variability in offspring number. Now I'm going to introduce a cost. So now species 1 lives slower, right? So it reproduces more slowly, but also dies more slowly. But also it's going to be paying some kind of cost. It's reproducing more slowly. So deterministically, that means that I actually have a stable fixed point up here. Okay. And so intuitively, or not intuitively, naively, I should say, you might expect that if I start off with 50-50, type 1 and type 2, I'm going to be going in the direction of the stable fixed point. But actually, that's not necessarily what you see, because depending on what values you choose, I can still see this kind of selection reversal. Okay, so here you have a cost of about 2%, which is fairly substantial in evolutionary terms, but a low population size of just 200 individuals. And here you can see that, so the black line is neutral, like kind of the kind of, what you call it, F1 line. The green line is what happens when species 1 and species 2 have the same B1 and B2, or the same R, I should say. So this is what you would expect normally. This is selection against species 1 because it's reproducing more slowly. But as I make species 2 kind of have these faster and faster reproductive dynamics, I start to see selection for species 1, even though it's actually reproducing more slowly, because it's reproducing more slowly, but with a much lower variance in its offspring number, and so it's selected for. The other type is reproducing more quickly, but with a high variance in its offspring number, which means it's going extinct more frequently. Does that make sense? Right. Okay.
Can you give an ballpark estimate what the difference should be to actually cancel this?
To make it
neutral, like, you know?
Yeah.
B5 is still B25 is still under the neutral line, right?
No, B25 is above it.
It's above it.
B2 equals 3 is below it, is just below it, and B25 is
Okay,
just
so
above
it's,
it. I
so
can,
it's
but not
twice
I can, but not off the top of my head. Cool. Okay, so and but obviously this is dependent on population size. So here are those. So basically, what I would expect just looking at that is I would always expect my fixation probability to be below this dash line. This is my fixation probability of starting with 50-50 of each type. I would always expect it to be below this black dash line. If I have exactly the same B1 equals B2 and D1 equals D2, I get this green dash line, which is in the region that I want it to be. But all the other lines have at least some region where they're above this black dash line, which says that they're being selected, that type is being selected for. Okay. And for this ridiculous type up here, where I have B2 is 100 and D2 is 99, even up to population sizes of 1,000 there, you can see I still have strong selection in favor of this the slower type. Any questions about that? Okay. Who knows like this sort of stuff in the background anyway, like these variants stuff? Okay, cool. So
look top of my head, but I mean I've seen similar.
Not surprising.
Yeah.
Okay, cool. Okay. So basically, demographic noise or stochasticity or drift, whatever you want to call it, combined with nonlinear population dynamics can lead to biased fixation probabilities that aren't apparent in the deterministic limit. Okay, so that's the general takeaway that is kind of model independent. In this particular example, we see that drift-induced selection favors slow-living species. And kind of the intuition here for me as a physicist is that these things always evolve towards a low noise state. So if I can pick a state and say that's the state where the noise is going to be lower, generally my rule of thumb is that's where the system kind of tries to evolve to. Yeah.
What is an example of slow living?
So you've got something that reproduces slowly but also dies slowly. Yeah. Tree, yeah. Reproduces slowly, dies slowly, or I don't know, a tortoise, probably. So they're slow. Bacteria live fast, and there's lots of them.
Tons of eggs.
Yeah, that's true. Okay,
But a whale.
a whale.
Yes. Yeah.
We'll get whales and elephants are one of the examples I'll bring up later.
Right.
Right. So, and this can lead to selection reversal in some selection reversal in some sense. Okay, so as in, I'm more likely to go in the direction that's the opposite of the selective. I'm more likely to see fixation in the opposite direction from the selective pressure. Okay, so this has been picked up on a bunch of different times by different people and in different contexts, but it's all just the same thing. Okay, now I can just quickly, just one more bit of time on this. What if, I'm going to change my conditions again. So what other ways can I rearrange this equation and still end up with something that's neutral? So the other thing I can do is I can say, let's Cij equal Ckj for all I and k. What that does is essentially gives me something like this. So importantly, the per capita reproductive rate of type 1 and the per capita reproductive rate of type 2 are identical, but now we've got these different competition terms. And now my phase portrait looks instead a bit like that. Okay, so essentially what I've done here is I've been able to change the carrying capacity. I still have this line of fixed points, but I now have this changing carrying capacity for one species relative to the other, right? Yes, okay. One way to view this would be costless cooperation. So type 1 here, when they're left on their own, they can grow to high numbers. Type 2, when they're left on their own, they can't achieve high numbers. And when there's a mix, type 2 benefits from an overall increase in the carrying capacity of the population from the presence of type X1. Does that make sense? It's cost. The fact there's a line of fixed points says the cooperators aren't paying any cost to do this. Okay, they're just increasing their presence increases the carrying capacity of the population. Okay, right. Fixation. Does anyone want to have a guess of what the fixation probability is? And we'll do it in terms of F1 again. So Q of F1. The reason I get, I'm just interested if, because I don't know. Half for what?
Like that's probably.
Oh, so it's a function of F1.
Probably I'm wrong again.
Well, it's weird. So the actual answer is it's just F1. Okay, and that's true for any value, any carrying capacities I choose. With this setup, the fixation probability is just F1. Okay, and that seems weird because I keep going on about this noise-induced selection. Because this is still not neutral in some sense, depending on how I interpret that. Because when a cooperating mutant turns up in a defecting population, its initial frequency is 1 over N2, where N2 is the number of individuals in the defecting population. Okay, 1 over some small number, because there's not many defectors, because they're all fighting over resources. In comparison, when I have a defector showing up in a cooperating population, its fixation probability is 1 over N1, where N1 is a large number, because the cooperators all live happily in their huge big community sharing invertes or whatever that they're doing, right? So this means that my probability of going from here to here is actually higher than my probability of going from there to there. Okay. So this becomes kind of clearer if instead of plotting this as a function of my frequency of F1, I plot it as a function of my frequency of the initial number of type 1. Okay. And I see that there's kind of this selective advantage. And just as before, this selective advantage can persist even in the presence of costs to cooperation. Okay, so the cooperators are benefiting from the fact that because there's loads of them, it's just probabilistically unlikely that a defector will be able to kind of overcome them all and invade. But they're going to pay a cost for their cooperative behavior, so some metabolic costs for producing invertase if they're yeast or something like that.
So you're now putting it as a function of initial number.
Yes.
The number is just the same as fraction divided. I mean, it's just scaled by a number. So how can a straight line be?
Because my fraction of my initial fraction of because F1 my initial fraction is N1 over N1 plus N2. So and now N2 is along this line, which is where invasion is kind of taking place. N2 is a function of N1, and N2 is given by something involving C1s and C2s and N1s. But that's basically what's causing this. And now when I add selection in, so I'm going to say, right, my cooperators are going to reproduce more slowly. That's going to create a stable fixed point up here. But this noisy effect is going to mean that there's still going to be conditions where, despite the fact that this is a stable fixed point, I'm more likely to transition from there to there than I am from there to there. Okay? Any questions? So what that means is, so I can plot, so the actual paper we talked about public good producers and non-producers. So producers in blue, non-producers in blue. Sorry, producers in blue, non-producers in red. Dash lines for deterministic. Deterministically, if I solve all my ODEs, my producers go to extinction and my defectors or my non-producers in red take over. But if I run a stochastic simulation of this, I actually see exactly the opposite behavior, right? And this is all embedded in it. So this is the other point, is that I kept going on about smaller population sizes, but all of these effects basically work in space. So if I have a meta population with a kind of relatively small number of individuals on each patch, then the stochasticity kind of kicks in, and this can work for very, very large populations. So this is a simulation where each of the little squares is going to be a patch. So this, I think this is 100 by 100. And I have about 100 individuals on each patch. And so this is, if it goes blue, that means that there's more producers on each patch. And you can see that even though if I solve my ODEs, the cooperators get driven to extinction, if I simulate the stochastic model, they win.
Any interaction between the patches or
Yeah, there's migration.
okay.
But migration is probably small. It's small.
Yeah, but it doesn't have to be. I can. I haven't got the plot of that, but I can show you. It doesn't have to be that small. So you can parameter. You can kind of. I say parameterize. You can roughly guess some of these things from experiments with yeast, and you can. It's kind of feasible. It's not completely ridiculous, but you do still have some parameters you could choose if you want to be kind to yourself, which. Okay, so how do I get out of this now? Basically, drift can favor a species with a larger carrying capacity. And again, in small populations, this effect can withstand deterministic kind of costs. And again, this can lead to sort of selection reversing. Okay. So any questions about that? Yeah.
One something about the previous figure. So you have this. So basically, you go to the deterministic fixed point.
Yeah.
But in the stochastic, you go to another fixed point.
Yes.
So I mean, to do deterministic analysis, you get your reaction rate equation. So you get your matrix A. You do your whatever stability analysis, and you finally go to that point. But in your stochastic case, so you get these second order equal correction terms.
Yeah.
So if I would do just my fixed point analysis on this equation, so my A plus my equal correction terms.
Yeah.
Do I then find that the stability has changed?
Yeah.
So you can explain the switch purely by looking at the second order correction terms.
Yes, but you don't get an idea. So you can just say when one is higher than the other. You can say when the noise-induced selection is greater than the. Sometimes there's also in general. Are you talking about in general in this particular example? In this particular example, there's basically just there's some things proportional to population size. That's my noise-induced stuff. Some things proportional to epsilon. That's my deterministic producers are rubbish at reproducing stuff. And they're going in opposite directions, and there's a flipping point there. And you can just basically say, okay, at that point, I'm more likely to go that way.
So you have to use your parameters sort of close to a specific point because your perturbation coming from the noise is small. So in some sense, if
well, no. So it's like that plot I showed you earlier. It's as the population size increases, this effect disappears. But it depends how. So if you increase your carrying capacity by, and it all depends on how much you increase your carrying capacity by. If you increase your carrying capacity fourfold, then it'll be a small population size at which this effect basically disappears. But if you're able to increase your population size 20-fold, then the point at which that
there was some sort of switch, but if you increase your population size, you will automatically be pushed below this switch and it disappears again.
So there's an interesting part there about the deterministic, but yeah, for actually some degree, if you can increase your even in a large population, if you can increase your population size substantially, you can see the selection reversal stuff. And importantly, this stuff really works in space. And actually what happens in space is this noise-induced selection gets squared. And I don't have time to explain why, but you get a squaring effect, basically. partly because, but in this particular case, it's because larger populations also produce migrants more frequently because they're denser. So you win twice. Yeah.
So I guess that the product of C and N is constant along this, along this line.
The product of C and N.
Yeah, you make a point of like, okay, when for N1, when type 1 is on its own, then N1 is large.
Oh yeah.
But I think that C1 times N1 is actually the same as C2 times N2. If you look.
Yeah.
Does your outcome actually depend on that neutrality of that product? Because if the Lotka-Volterra is of course a very specific system.
So actually, the example in that paper isn't Lotka-Volterra. You can show this works for arbitrary sort of uptake rate of you. So you can add a resource in here.
Okay.
So it's then the example I look at in the paper. You've got you've got invertase, say, and you have one of the types produces invertase, the other doesn't. You've got a resource, and you can add all these extra details in. So what happens is that in terms of if I just want to think about species 1 and species 2, I end up with that. I don't end up with a straight line. I end up with some weird shape depending on what I've chosen for my uptake rate function and things like that. Everything still works. The only place that all this mathematically I have a nightmare with is where the population crashes, where there's no cooperators. So mathematically, if you have 0 population size when the defectors, when you end up with defectors, then all the maths breaks down.
Okay.
But you can look at all those generalizations, and so this is all this is generalizable. So with a case, it's kind of as a case in point, I'm going to get rid of it. So this is all kind of a bit abstract. Oh, and this has also been covered in different forms by different people. Okay, so I did some work on this, but Oscar Halacek, basically this turns out to be the same model in this paper. Baharam, Huchman, Mendeza, and Marcel Vlad have also looked at something differently, but with a fixed population size. And then more recently, David McLeod and Troy Day and Shakara Bhatt, a bunch of different people have kind of looked at similar variations on a theme, basically. So there's a lot of, basically, I suppose those these slides with these papers, what I'm trying to say is there's people kind of looking at something through, some of this is basically kind of well-known stuff, but it keeps popping up in slightly different areas. And if you know what I mean. That makes sense? Yeah. Okay. So here's another area it pops up in, sex determination systems. So we have our males are heterozygous. Okay, so Y is our sex determining chromosome. So females are XX and males are XY. It doesn't have to be that. So basically, if you end up with a Y chromosome, typically in mammals, you end up as a man or a male. In birds, it's the opposite way around. So there, males are homozygous. They have the same chromosomes, WW, and it's the Z. It's the sex determining gene. Any questions about that? Okay. So if I look across the tree of life, birds and mammals are actually kind of weird because they've just ended up stuck in these two systems, and there's reasons for that I don't have time to talk about. But actually, amongst most other species, there's some variation in the sex determining system. Okay. And most interesting is that amongst amphibians and reptiles and fish, there's lots of examples of species that do a bit of both. So in this Japanese wrinkled frog, Ana rugosa, in the north of Japan, it uses this system. So the males are heterozygous. In the south of Japan, it uses the bird-like system. The females are heterozygous. And in the middle is a bit of a mess. Okay? So, in terms of understanding the evolution of early sex chromosomes, we want to understand this. And this is the kind of model. This is a book from Bull and Charnoff in the 70s. So, I have something that's mammal-like, and then a mutation comes along on the X chromosome, call it X prime, and this is a feminizing mutation. And we're imagining something here where the Y, so do everyone know about the stumpy little disgusting Y chromosome? Yeah, so we're imagining not a system like that. We're imagining a system where this hasn't degenerated, like the mammalian Y chromosome or the bird Z chromosome. So early days of sex chromosomes, something like this frog, a mutation shows up and it's a feminizing trait on the Y chromosome. So it's overriding the Y and saying anyone who carries X prime Y is going to develop as a female. Now I have a female X prime Y, and this female X prime Y can mate with males that are XY. And if an X prime Y mates with a male XY, that can produce offspring that are X prime X or YY. Okay? So you can see why this transition doesn't happen in species with degenerate Y chromosomes, because this YY type ends up dead. But I have an absorbing state here, which is male heterogamity, like mammals, and an absorbing state here, which is female heterogamity, which is like birds. And so I might ask how these transitions happen. So if I transition from this stable state to this stable state, I'm going to call that transition A. And if I transition in the other state, I'm going to call that transition B. I can basically write all this out. I get 5 ODEs or 5 SDEs or 5 whatever. I can't plot that out very nicely, but I can plot that out just as a definity diagram in the female plane. I get something like this. A load of fixed points emerge. I have an absorbing state up here, and I have an absorbing state down here. I have a line of fixed points, the center manifold between them. I can use all the same mathematical theory I talked about before. Naively, you might expect that transitions are the same in both ways, but actually, when you mathematically go through all of this, you find that transitions are more likely in the A direction. And that actually is true as I add more mutations. So here's another mutation shows up. So I end up going from there to there. That happens with higher probability. Now I'm in this state. Actually, a mutation can show up. It's this Y prime, and that's a masculizing Y prime. So now what that is doing, sorry, not masculizing, it's a feminizing. No, it isn't. What's it doing? It's doing something. What does that mean? Anyway, I can end up with the same behavior repeating, where now the Y prime is evolving a dominant trait to basically try and get back control of the X chromosome. There's more transitions, but essentially I can show that A equals C and D equals B. Pardon?
The mutation is making it male, right?
Yes. It's making, yes, it's recapturing, that's it. It's recapturing X prime Y, and it's making X prime Y prime now male again. Exactly, yeah.
If I understand correctly, we do secret assumptions, like with X prime, we make an assumption that is feminizing.
Yes.
But then what with YY, we are not make another assumption that YY is male and not like another third thing.
Yeah, that is an assumption. There is a, so actually there's even more assumptions in this. So this is the simplest. We go through 2 alternative models.
Yeah, we assume that one of the labels will always
apply. There's another model of this that's more complicated, involves sort of 8 variables, and we do that in the paper. But then there's other models that don't look at everything in such a binary way. But yeah, they get more and more, you need more and more variables. So the short reason is reduce the number of variables. But anyway, it turns out this prediction that there's this bias to go in this direction is kind of borne out by some empirical observations, which is basically that it looks like in the early evolution of sex chromosomes, there's this consistent pattern where initially the sex chromosomes are evolving to always try and be dominant to each other. So they're all moving in this direction. And they're not evolving to be recessive, which is this backwards direction. And this pops out of the model really easily just by adding noise. Okay. And that was published in this paper. But it's quite nice because it's a very kind of simple explanation for some observed empirical pattern that doesn't require big, heavy, lots of machinery. What time is it, because I can't see a clock.
So 5
past 12. Okay, so I'll just try and quickly. And did I start at 11?
Yeah.
Okay, so I'll quickly.
relaxed.
Just very quickly then. So basically, my point is that the examples at the beginning were mainly just toy examples. Just I know that they're daft, but they're more to try and give people a feeling for what's going on. The thing is that this stuff appears in more realistic scenarios. And then the last thing I kind of wanted to sort of tie it to was models of growth, coalescence, and fragmentation. And this is kind of ongoing work. So my old postdoc boss, Karina, published this paper 10 years ago. And this was basically just trying to write down some equations for basically trying to use this modeling framework about staying together and coming together to try and visualize the different forms of primitive multicellularity that we observe. So things like the bulbocene algae, you have a daughter cell, the daughter cell stays with you, and you keep reproducing until you form this kind of like primitive multicellular thing where all your daughter cells have stayed together. That's staying together. And the alternative is coming together. So that's something like dictyostelium, where separate, unrelated cells come together to form a primitive or not so primitive multicellular thing. Okay. And I can also think about things like these pictures from Will Ratcliffe's group of sort of snowflake yeast, which is staying together. This is my offspring are sticking with me due to some extracellular sticky membrane thing that I'm producing. Or I can get flocking, so cells coming together. And the important thing is, or not the important thing, but an important thing about thinking about things like this, staying together happens slowly. The rate at which I can form something multicellular and staying together is limited by my growth rate, because I can only grow as fast as I can produce daughter cells. Whereas coming together, I can come together very quickly. I can just take a bunch of unrelated cells and we can form something together, but we're likely to pay the cost that we're unrelated, and we might have differing objectives in terms of what we do or what we get from the system. Right? Any questions? Okay, I'm leaving out all the interesting stuff. I'm just going to look at this same thing of, do we get this same noise-induced behavior in this system? Okay, and the spoiler is we do. So what I have here is I've got unicells. This is basic. I've added a resource here, but this is basically, I could just rewrite this as Lotka-Volterra if I wanted. But I have X's are eating a resource and reproducing. That's just the same as before. I'm adding resource into the system to keep things going. I have death of of type X, and I also have competition, but I just remembered I forgot to add that in, right? So there's a carrying capacity. Oh no, I don't need the carrying capacity. That's it. Sorry, the resource is setting the carrying capacity. So there's a carrying capacity enforced by the fact that all these things are eating the resource. Okay, so A1 is the reproductive rate of single cells on their own. G is resource growth. D1 is some sort of cell senescence, and M1 is some sort of extrinsic mortality, which I've called two different things for a reason we'll see in a minute. Any questions about that? Okay. And then I'm just going to look at staying together, but everything generalizes. So I've got these multicellular units. So what's going to happen is at rate AI, multicellular units of size I are going to produce a daughter cell. And with probability Q, that daughter cell is going to stay with the parental complex and grow a bit more. And with probability 1 minus Q, it's going to detach and form a new single cell or propagule. Okay. And I also have cell senescence. This is individual cells within a complex can die or fall off or something. And I also have extrinsic mortality, which is the whole thing dies. Okay. So those are my parameters. The Q is like a stickiness parameter. Any questions about that? Okay. Does anyone have a sense for what the neutrality, in a deterministic setting, what the neutrality condition should be? So what is the condition where neither of these wins?
Both of them are dead.
That is true. So if they both die, there's nothing, but if they're both present, you have a feel for it.
Somehow this MI should relate to the shouldn't MI's scale with I?
That's interesting. So no, but close, but weird also. So the neutrality condition turns out to be, if I want to set this out, so birth rate, so the scaling with I is really important for AI. So basically what this says is that two cells should be picked to produce a third daughter cell twice as quickly as an individual cell should. Okay, so the rate at which a clump of cells produces a single daughter cell is proportional to its size, which kind of makes sense.
What's your B?
Pardon?
What is your B?
Just B, just a constant B.
Oh.
Sorry, yeah, I haven't defined it anywhere. Sorry, this is just so this A1 will be B, A2 will be 2B.
Okay, so AI should be proportional to I. Okay.
Yes, so basically if I have a clump of 10 cells, I pick them to that clump of 10, because there's 10 cells, I'm basically saying I'm going to have to pick one of yours to produce a daughter cell. So the per capita rate of cell production is basically independent of involvement in a complex. Doesn't matter that I'm involved in a complex, I haven't improved my per capita rate of producing daughter cells. Same for senescence. Okay, so I should be picking these clumps of cells. An individual cell should be dying within that. So if I have a clump of size 10, I pick that clump 10 times as much because there's 10 times as many cells that need to be knocked off potentially and killed off. But weirdly, mortality should just be, the extrinsic mortality should just be flat. So this is weird in some sense, or maybe counterintuitive. So basically the same per cell reproduction rate and cell senescence rate should be present, but my extrinsic mortality per individual should be flat. Okay, so this is weird because there's something here which is about biomass and something here which is about the nature of the individuality in this system, and not individuality is probably a loaded word. Something about the shared fate of these cells. On one level, you're looking at something in terms of biomass, the other you're looking at something in terms of, I think this is non-obvious, right?
I actually, yeah, this
Go
is
on.
a point of discussion. Well, so your DI, so in the multi-cells, your DI is not a per capita rate. Only it is it becomes a per capita rate if you choose that it's proportional to I. And the same holds for this AI. So, you know, in the top row, everything is per capita. A1,
Yeah.
D1, M1 is per capita.
Yeah.
In the multi-cells,
yeah,
the MI is still per capita because if you know every individual has
experiencing the
the same mortality. So I would actually say that the definition of your AI and your DI in these multi-cells, they differ because they are not per capita rates.
Okay.
And that's exactly why you say like, okay, they're neutral if you choose them such that they per capita correspond to the top.
Okay.
Right, something like that.
Yeah. But you're happy with this?
Yeah. So
maybe with the maths.
The weird thing is not the MI. The weird thing is the AI and the DI, which for me.
Okay. So just to give you a bit of orientation, I'm just going to plot like the stationary distribution of what densities I see. So if I make things a little bit sticky, I start to see some cells that are some complexes, some clumps of cells that are getting bigger. If I make things really sticky, then I start to see really big clumps of cells. So what happens? No, I get noise-induced selection again. And actually what happens is I get a crash in my fixation probability of these multicells. Noise is selecting against the formation of these large complexes, and it actually crashes into 0 above Q equals 0.5. And so what's happening is, as I produce bigger and bigger clumps of cells that are vulnerable to extrinsic mortality, I'm really amping up my noise. There's a probability per unit time that I just knock out a huge amount of the biomass from this population in one fell swoop and crash the population essentially.
This depends on you then.
Yeah.
This depends on the presence of some extrinsic mortality, yes, which is why I find it weird. So if there's just cell senescence, then no, but if you have extrinsic mortality, then there's this crash in.
Yeah, because it like links the cells together, right? They're fate.
Yes, exactly. Yeah. And this is both intuitive and kind of not at the same time, because like depending on what mood I'm in, I kind of think this is obvious or not. I don't know.
But the Q was the probability to stay together, right? So if you, by default stay together, Q equals 1, and so then your population immune.
Yes, exactly. Yeah. That makes total sense, right? You end up with this just huge, but exactly in that limit makes total sense. You just end up with one cell, and then you kill it off.
Mu hits, and then everybody.
Right. Excellent. So, you can see, and this is basically a function of this mu term. So if mu is, if I get rid of any extrinsic mortality, I just get what I expect. But if I amp up my extrinsic mortality, I start to see this region where everything dies. Okay. So basically placing more biomass at risk of extrinsic mortality is this risky strategy. This is, I can change this maximum complex size. So if I make the maximum complex size smaller, if I basically say, okay, well, in that mathematical model, I can just grow massive, but let's just say I can't grow massive. Let's put some hard limit on it. That kind of dampens things down a bit, but I still see selection against putting all my eggs in one basket, essentially, by creating these large, these fewer individuals, essentially, because if I grow bigger, then the population can maintain fewer individuals. If a population with fewer individuals is more vulnerable to extinction. So who goes extinct first? Whales or bacteria? There's a huge amount of biomass in these whales, but there's only a few individuals. It's very easy to kill them off by random events. I can also add some sort of fitness landscape. So this is me saying, okay, this is just a really simple one because obviously I can choose whatever fitness landscape I want. But this would be something like if I was looking at, so algae can evolve to be multicellular to escape predators. Okay, and there's experiments in Plume doing this. So the idea is you get some critical size, and this other little microorganism can't eat you anymore because you're just too big and it can't get its mouth around you. And then you can reproduce more because you're not getting predated on. And so if I do that, what I can see is that sometimes I'll see this, I'll get selection for this kind of stickiness, but it might be after some sort of fitness sort of valley, essentially, because before I get to that perfect big size, there's a load of intermediate stages where what I'm doing is I'm making myself bigger, opening myself up to risk by being eaten by the predator, but it can still eat me, but it's also getting me all in one go. Okay.
So what do you assume about the extrinsic mortality related to this complex size? Because this dip, it might depend on like how your assumption on how your extrinsic mortality risks.
Yes. Yeah. So I've just got it flat here again. So this is my reproduction. So yeah, you're right. I haven't got separate predators in here. This is just me kind of motivating. This is exactly the same model as before. You're right. There'll be feedbacks to the predator population, and I've not done that. I've not dealt with them. I'm just kind of, this is me. This is all ongoing. So now this is published. This is all just me being like, kind of maybe. I don't know. Yeah. And also trying to get some feedback off people. Oh yeah, okay. So that's basically it. Is this? I thought this was kind of weird. So noise generates selection against processes that concentrate biomass in pure individuals with shared risks. Okay, so that seems weird in some ways, or some of it seems surprising, but it's kind of not surprising if I think about something like terrorist cells. So it's weird for me to think about when the stochastic option is obvious and non-obvious. If I'm organizing a cell, I make my cell small so that when a cell is discovered, I don't lose that many of my terrorists. I don't know what you call them, but my network, right? So this kind of stochastic process, that works non-intuitively. So sometimes the stochastic understanding is kind of what makes sense to us. And kind of what I'm potentially interested in is kind of weird, this kind of death of individual cells within a complex, so senescence, in my head, but we can have a chat about it, delineates the model of this staying together from some discretized model of cell growth. So that model that I showed earlier, what's the difference between this? I basically just have little lumps that I'm adding together, and I'm saying 1 cell, 2 cell, 3 cells, 4 cells. But that is also a discretized model of growth, where I have cell of size 1, cell of size 2, whatever. I'm kind of interested in which of those two, or how those two models differ, okay? Because they're exploiting the same. If I forget about metabolism and I just think about advantages of being big in an ecological sense, I could either just produce massive cells, or I could produce little clumps of cells. And what's the difference? And so this is the fisherman. Do people know the fisherman's eyeball? The biggest unicellular organ. So that's someone's fingers. So this is a huge unicellular organism. Okay, so I can grow pretty big. Why do I need to go multicellular? And I'm kind of veering in that direction because of looking at this model, but now kind of thinking more about the biology.
I think this is also Elon Musk's rationale about going to Mars. He's saying that we're just a huge clump of biomass on Earth.
Yeah.
We're very vulnerable for a meteorite.
Yeah.
So we need
you have some sort of shared fate.
We need to spread our biomass.
Yeah. So this is. I'm interested in these kind of weird noise-induced questions, but now also this question of when should I grow? If I just looked at if I forgot, so there's lots of answers to this question of when to grow big versus when to grow multicellular. If I think about kind of cell energetics and things like that. But I'm wondering about from the other perspectives, what kind of makes sense. Anyway, this is ongoing work with Karina and one of my students, Becky. But basically, kind of going back to those questions, so when does stochasticity lead to qualitatively different behavior? So I've told you a bunch of different stories of selection reversal, but there's lots more out there. And sometimes it gives you these parsimonious explanations, maybe evolution of cooperation. It's quite kind of straightforward to just add in the selection reversal. There's lots of other examples of where this is the case, though. So I've got some work on fungus sexes. So in fungus, you can have more than 2 sexes. In Schizophilum commune, there are 23,328 different sexes. You might wonder what controls that number. So I've done some work on that. When can noise be leveraged to tell us more about a system? So I've got some work using these noise terms to infer maybe ecological interactions or something about rates of sexual reproduction in the population, and things about cell size and cell fusion in facultative sexual reproduction. I've got lots of work on that as well. So those are some of the things I haven't been able to talk about. But yeah, if anyone is interested in those types of questions, I'd be happy to chat. Thank you.
Yeah, because just wondering something about these. We showed this with the sex chromosomes.
Yeah.
And then these mammals are sort of a rare old case where we have this degenerate Y chromosome. Can you in your models sort of find a pathway where this makes evolutionary sense? That we should sort of put down this track of having this degenerate Y chromosome.
Yeah, so do you know the story? Are you asking what the story is of why? Of why that happens, or are you asking?
Yeah, can you see it in your model? Like, is there
Oh, no, you can't see it from the model, but you can kind of see. So basically, the model you can see in the model why.
So you assume here that
No.
the X and Y are both healthy, functional.
Yes.
Yeah.
So basically, it's because you evolve to, in order that you basically, because this is going to be, you get recombination shut down along that Y chromosome, because you otherwise you end up with, say you end up swapping bits of your masculizing Y trait onto the X chromosome. So basically what happens is whoever this Y here or the Z there, because it's the one that's on its own and it's controlling the sex, it doesn't want to have recombination going on, because then it'll pass its so in the case of the Y chromosome, it's masculizing traits over to the X chromosome, which means that you'll end up potentially with unfit daughters. That's the kind of idea. And so there's a selective pressure to shut down genetic recombination, so shut down the crossover events on the kind of sex determining chromosome, which for us is Y and for birds is Z. Once that happens, though, because there's no recombination, mutations build up, and so you basically end up with the Y chromosome building up all this crap, like these deleterious mutations, which it can't shuffle it away. And because of that, the only way to get rid of them is basically just to chop bits off periodically and hope that you chop off a bit with the deleterious mutations in. And you just keep doing that over and over again. Eventually, you've got nothing left. And that's why that gets stumpy. As that gets stumpy, though, and starts losing function, that's essentially why. So the longer you stay in one of these states, the more degenerate this sex determining chromosome is going to get. It's the same for this Z over there. So the idea is you hop back and forth between these states of the sex chromosome. So initially in the evolution of sex chromosomes, you fight with them trying to take over from each other. And then at some point you spend a bit too long in one of those states and this chromosome starts getting rubbish. And then once that starts happening, that's kind of like a pit you can never escape from, is the idea. And before all this, there's genetic sex, temperature, sex determination, and all sorts of other things. And yeah. So these transitions are kind of fun to look at.
So why does X prime invade here?
It doesn't. What do you mean why? What's the
Why
noise
would
story?
you get the mutation X prime?
Just mutation.
Yeah. But you can lose it directly, right?
Yes.
Right?
So
This is completely neutral.
just yeah, completely neutral. So it just shows up, and it's just we're wondering if is it going to be is it going to drift to fixation basically, and weirdly it's more likely to drift to fixation that way. And I haven't got any intuitive. The only way you can show that is, like for me anyway, is with the maths. And we spent a long time trying to come up with an intuitive explanation, and they all failed on some level. Like we came up with things that kind of made sense, but then you test them out and they weren't true. So, but mathematically, you're much more likely to go that way. And that seems to match what we see. And it's robust to the choice of model. So we looked at more complicated versions of this model, and the same thing seems to come up again and again. There's a noise-induced bias for these dominant mutations on the sex chromosomes.
Does it make sense that the whales have to be sort of slowly reproducing because they're multicellular, like extremely? So they've got, the vulnerability to death because they're so big.
Yeah.
But then you should counteract that by being slow-lived and slowly reproducing.
I love that idea, and I'd love to, because I don't know if that idea is mad or not. So yeah, I thought about this. So if I now try and draw neutral, like you were saying earlier, what's the selection strength I need to counteract this noisy stuff, right? And then at some point I started thinking exactly this: what is the if I want, I've got all these noise terms now, so I can grow multicellular, I can change my carrying capacity, I can reproduce more slowly, and all those things give me a noise-induced advantage. So if I leave my system completely unanswered and just say, what does noise do? Well, I get the result that I should have a species that's unicellular with a very high carrying capacity, but reproducing extremely slowly. or not slowly, it's with extreme, with a very low variance offspring number. Okay, so that kind of makes sense, but also doesn't. So I was looking at what are my neutral surfaces within this, because I've basically got growth, I've got growth and reproduction rates and things like that, and I can look at what is this neutral surface where they all cancel each other out, and I still end up with something that's everything's neutral, and it looks like you get something that kind of looks like life.
Yeah, because I mean, you can either go the one way.
Yeah, you
and
can be
go
big and
different
slow. Yeah,
or you can be small and fast.
yeah, but then I don't know if that's not obvious. So I was like, oh, is this interesting? And then I was talking to someone and they were like, well, of course, big things are slow because they have to grow big. And I was like, oh. And then that made me less excited about it. So I don't know if it's interesting.
So one of the limitations of this whole framework is, of course, is that it misses individual development throughout life, which is pervasive throughout the tree of life, and which is always ignored in Volterra.
Yeah. So that's why that's why. So you can build a lot of this into this like kind of that sort of this ontogenetic kind of approach.
Yeah.
It's kind of what I'm interested in this multicellular stuff. We're trying to get move into is something that says, okay, what happens with ontogeny plus noise?
Yes.
And what can I start to see these effects in those kind of models? Basically, it's kind of one of the things I'm interested in, and I'm not sure because they're kind of continuous. I need to start thinking about SPDAs because you've got these continuous growth of individuals, but then you've got this discrete nature of the individuals making it up. So, but it doesn't make sense necessarily to think in terms of biomass, but it also doesn't make sense to think in terms of these crusty individuals. And so, my halfway point is thinking about these discrete.
There was another remark you made about, like, I forget about maintenance, you said, right? So
yeah,
I think that the role of maintenance in these kind of dynamic and population dynamics is terribly under represented.
Yeah, because so this is what this is
all models assume that even if there's a little bit of resource in population start to grow or reproduce, which is silly because you need that food if there is little.
So all the
sustain yourself, you cannot do anything.
I've not seen, but I might not just be looking in the right places. A nice model that looks at these kind of stochastic individual based stuff, but does it in a nice way that has individuals with some kind of nice growth and maintenance costs and all these other things in some kind of consistent way. Because generally kind of what you do is you're like, fox eats rabbit, fox has baby fox. And you just kind of, well, not even baby fox, fox has fully grown adult fox after eating them. So you get these nice noise dynamics, and you get, but they're kind of nonsense. And then you try and go the other way, and you're like, okay, how do I capture this in such a way where I have a continuum of foxes that are all, don't know why I pick foxes, but yeah. So based on these results where I have this biomass selection against, that's why I was kind of interested in, okay, what happens when I've got a growth model? Do I actually see this selection against growing large in some continuum model? Okay.